It is my distinct pleasure to introduce Dr. Raymond Deshaies, Holy Cross Class of 1979, who joins us as Professor Emeritus from the California Institute of Technology. After Holy Cross, Dr. Deshaies earned his bachelor's degree from Cornell University, and working with the Nobel laureate Dr. Randy Schekman, he earned his Ph.D. in biochemistry at the University of California, Berkeley. He did further postdoctoral training at Berkeley and then with Dr. Mark Kishner at UC San Francisco. Dr. Deshaies took a career-long faculty position at Caltech in 1994, rising through the ranks and earning tremendous accolades. Among these are his designation as a Howard Hughes investigator and election to the prestigious American Academy of Arts and Sciences and to the National Academy of Sciences. Dr. Deshaies studies how cells stay healthy by controlling their proteins, how they are made, how they work, and how damaged or unnecessary proteins are broken down. This process is essential to life, and when it goes wrong, it can lead to diseases like cancer and neurodegeneration. His discoveries have fundamentally reshaped an entire field of modern biology and has directly influenced new approaches to drug development. Dr. Deshaies is also an entrepreneur, starting companies and doing businesses in drug discovery. Until recently, he served as a senior vice president of research and development at Amgen. Please join me in welcoming back to Holy Cross High School a crusader and scientist, Dr. Ray Deshaies.
Okay. Wow, thank you. It's really a thrill to be back here. I was last in this room in 1979, 47 years ago, when I walked across that stage and got my degree, my diploma. I may be old, but I still got game, and I'll challenge any of you in this room to mountain biking. I'm going to give you, this is going to be a very unusual presentation. I don't think I've ever given one like this before. I've given a lot of presentations in my life, where I'm going to reflect somewhat on my growing up and then parts of my career, and what were the things that were important for me in my life. Even though I feel my life still has a long way to go, I hope, it's as good a time as any to reflect on that. So this is where I grew up, that X right there, 76 High Street on the, in the north end, on the east edge of the north end there. And it's kind of an interesting place. I don't know if any of you have ever been on High Street, but yeah, lock your doors if you go. This is the house I grew up in, 76 High Street, on January 6, 2013. I was there on that day. My dad lived in that house until about 2006, when he was 91, or 2005. And then he lost, he was 91, he lost his license because he was starting to have vision problems, and he really couldn't live there anymore. And so he moved to Boston to be near my brothers who were living in Boston. And the house, when he left it in 2005, it looked beautiful. He maintained it, even though it was in the hood. He took really good care of it. Somebody bought the house from him. Obviously, it was a slumlord that bought the house. They ran it downhill, and this is what it looked like on the day of my dad's funeral, when I came back to Waterbury for his funeral. So he died on January 1st, 2013. That on the right-hand side there, you see on to the left of the house, there's a boarded up window. That was my bedroom. So that's the room right there. That was my bedroom that's boarded up there. That was our door to our apartment. We were on the first floor. My parents had bought this house right a couple of years after getting married. They lived in Prospect first for a while, in a little cabin that my father built with his uncle. And then when they started having children, they needed more space. They bought this house. And so, and this will factor in later on, they rented out the 2 upstairs apartments, and we lived on that first floor apartment. This is what the house looked like 8 years ago. Literally, there were homeless people living on the 3rd floor, lighting fires to keep warm during the winter, and one of those fires got out of control and the house burned down. I haven't been back to High Street since, but I think the city tore it down. I hope the city tore it down. I saw houses burned down, like literally across the street from me, for the same reason when I was growing up. And if you will go down High Street now, I would say at most half of the houses remain because they've all suffered the same fate. This is a picture I took of my parents in our yard, in our backyard. It's actually a side yard, Jerry and Rose Deshaies. This is probably in 1997. My mother, at the time, she had contracted a very rare form of lung cancer called a Pankos tumor. And she was still in very good shape at this time, but she died about a year after I took this picture. This is my dad. My mom was a homemaker. When I was born, she didn't even speak English yet. They were both immigrants from Quebec. My dad came with his family when he was 12 years old. My mother came when she met my dad at a funeral in Canada after World War II, and they fell in love, and she came back as what at the time was called a war bride, which enabled GIs to take women that they met in other countries back, and they would get citizenship. So she came at 24 years old, and she didn't speak any English whatsoever. And she really didn't start speaking English until I was born when she was 40. So the first 16 years she was in the US, she just spoke French Canadian. They were members of St. Anne's Parish. All her friends were French Canadian. And she only spoke to me in Canadian, in French. And I refused to respond to her in French. I would only speak to her in English, and she would get mad at me and say, "Why don't you speak French to me?" Mom, I'm not French. And so my dad worked at Scoville's. It was interesting because in the drive back yesterday, some of the students were asking me, what was Scoville's? Scoville's was this enormous factory that stretched from the east end of downtown all the way beyond Hamilton Park, where there's now a huge mall. It stretched along Route 84 and dominated the city of Waterbury. My dad worked there for his entire career until he retired. This is in our first floor apartment. This is me. I'm actually really angry at this time because the 7th game of the World Series is on, and it's a crucial time in the game, and they decide they need to take a family photo, which I was not totally enthusiastic about. This is my brother Roger, who's passed away. This is Richard. He was a member of the class of '72, the first class at Holy Cross at this high school. And you could tell it's 1972 because look at that hair. And the other one's my brother Bob on the right. This is my yearbook photo. I mean, come on, look at that hair. This is like 1970s, big time. Here's one of my famous classmates, Mark McDermott. And he actually lived just 3 or 4 blocks away from me. He lived at the corner of Walnut Street and Cherry Street. Some of you may know him more as Dylan McDermott, who went on to become a Hollywood star and won awards for his role, particularly in The Practice. A great show. I used to watch it all the time. But I got a Dylan McDermott story. And so we rode the bus together for 4 years, the school bus. We didn't go. He went to St. Mary's, right across from St. Mary's Hospital. I went to St. Anne, which was a little bit further down, near St. Anne Church. And so when I came to Holy Cross, you had to wear a suit or sport jacket, shoes, and tie, okay? Now, when I went to St. Anne, you had a similar thing, but it was a uniform, right? So everybody wore the same thing. And so maybe you would have 2 or 3 copies of it, but every day you look the same. So when I start coming to Holy Cross, we were poor. My family did not have much money. And my parents take me to Anderson Little, which was in the Naugatuck Valley Mall, and we buy a suit, okay? A suit and a tie, one suit. And like a brown corduroy suit. I am not joking. A brown corduroy suit. But that was like the height of fashion for me. Remember, this is like 1975. And so I proudly wear my brown corduroy suit get on the bus Monday, get on the bus Tuesday with my brown corduroy suit, get on the bus Wednesday with my brown corduroy suit. Finally, I'm getting on the bus on Thursday with my brown corduroy suit, and at the next stop, because Mark was the next stop down, the last stop on the line, Mark gets on, and he comes and he sits in the bus seat right in front of me, and Mark could be a wise guy. And he just looks at me and he's like, "What, you only have one suit?" And I immediately respond to him like, "Well, of course I don't only have one suit." And he goes, "Well, you've been wearing the same suit every day." And I said, "Well, it just so happens that I really like this suit." And I got home that night and I go to my parents. I'm like, "You need to buy me another suit, like now, immediately." And to my parents' credit, they bundled me in the car. We drove to Anderson Little. We bought a sport jacket, a couple of different sport slacks. And the next day I showed up on the bus. And when Mark came on, I said, "See, I told you I had more than one suit." Now, of course, we have other famous members like our mayor, who was just here, Paul Pernaruski, who was also one of my classmates. So a lot of amazing people coming out of this place. Now, I'm just going to go through some key sort of life pivot points for me and describe why they're important for me. And so this is very personal. It may resonate with you, it may not resonate with you. I'm just going to tell you what was important for me. So a key life inflection for me is when I left here, going to Cornell University, where initially I went to major in horticulture. And that came about because of a mentor, a completely unexpected mentor, who was one of the tenants in my parents' house. She lived on the 3rd floor, and she had come to Waterbury in a program that now is similar to what's called AmeriCorps, where people graduate from college and then they go into a service program to do some sort of service. And the service program here was putting people into the inner city, into very difficult neighborhoods that have a lot of problems, doing various social work programs. And so she came, lived in the 3rd floor, and my parents, these were 2-year programs, and they had 2 cycles of young women there, and they became close. Both groups of young women became close to our family, would have dinner with us on occasion and stuff, and we're still in contact with the 4 of them that participated in this program over those years. But Dorothy, her name was Dorothy Hall, she never left. When the program ended, she stayed in Waterbury. I think she initially was in the program from 68 to 70, but then she stayed. I don't think she left until probably the mid-80s. So when I was growing up in the 1970s, I would go up into her apartment and hang out with her and talk with her. And she mentored me. She would give me books to read and all sorts of stuff. And when it is time to apply to college, I was probably going to go to UConn because that's where my brother Roger went and my brother Richard went. And I figured I'll go to UConn. I had no idea what I wanted to do. And Dorothy said, "Well, you're really into plants," which is true. I was growing these crazy plants, spending a lot of time. I was really into growing plants. And she said, there's a school I think you should really go to. And I said, "Well, what was that?" She said, "Cornell." I said, "I've never heard of Cornell." She said, "Well, they have the best college of agriculture in the United States, like rated #1. You should go to Cornell and major in horticulture." And I was like, "Oh, okay." And then I looked up and I realized Cornell was an Ivy League school. I said, "Well, I'll never get in. And if I get in, I could never afford it." And she's like, "No, don't worry about that. You'll get in, and if you get in, you'll get a scholarship." So I applied to Cornell. I applied to UConn. I got into Cornell. I got a scholarship. And I went. And I went to the College of Agriculture and Life Science, which is run jointly with State University of New York. And I majored in horticulture. Now, that lasted all of 1 semester. The first semester I had to take some, a course in my major area, which turned out to be pomology, which is the science of fruit tree production. We had laboratories out in the orchard. The people who were in the class actually, grew up on farms in New York State that they were going to take over, when their parents retired. They're going to take over the family farm. They show up in their bib overalls, and I was like, this isn't my crew. And so I started moving gradually in the direction of small biology, but not really knowing what I wanted to do. But for me, the key thing about going to Cornell was sort of the diversity of people that I met. There were people from all states at Cornell. There were people from many different countries. There were people that came from many different backgrounds. And it was just the range of experiences that the people I knew, the range of ambition, the range of visions of where they wanted to take their lives. And it was just very eye-opening for me. It was a tremendous, fantastic experience. And I met many people there that I remain in contact with today, including my wife. It was really a fantastic experience. Now, what I did there, as I said, I started gravitating more and more to small biology, biology at the level of cells and smaller. And, but I had no idea what you do with that. And growing up in Waterbury, there was sort of no real role model for thinking what does somebody who major in biology do other than become a doctor. So I became pre-med, officially, technically, pre-med. But then, I had not done quite as well as I might have hoped in a couple of chemistry courses. I think I might have gotten a B- and a general chem course and a C+ and an organic chem course. And the whole thing was oh my God, if you want to go to medical school, you need to be straight A. I didn't volunteer in any hospitals. And so, it's junior year, and I'm thinking well, I don't know how competitive I am as a pre-med, and I don't know that I'm really that interested in being a doctor anyway. It was just what do you do if you major in small biology, if you don't become a doctor? I had no idea. I made the decision to stay at Cornell after my junior year because I'd come back to Waterbury after freshman and sophomore year, and in particular after sophomore year, I had a factory job in the summer, which I absolutely hated. It was awful, and it convinced me I never want to do this sort of thing ever again. So I decided to stay at Cornell. I got a job in a research laboratory, and what I found over that summer is I absolutely loved doing research at the bench, doing biochemistry research. And I liked the process. I liked working with my hands. I liked thinking through the problems that come up when you're doing experiments. And I basically decided at the end of the summer, I'm going to apply to graduate school. I mean, literally, entering senior year, the applications are due in a couple of months. You have to take the GRE exam, all this stuff. I decided at the last minute, I think I'm going to try to go to graduate school. All right, so I tell my parents, my brother Roger, they get really upset with me. They're like, you're throwing all this away. You could go on to medical school. You could be a doctor. Doctors are important. It's well respected. And what's this research thing? And, they just were having none of it. But I figured, it's my life. It's not their life. I'm going to go to graduate school. And, I reasoned that because I had no idea what you would do with a graduate degree, I figured, I'm going to go to the best possible graduate school I can go to and just see if I like it. And so I ended up applying to Johns Hopkins. I applied to University of Wisconsin-Madison, which had a great biochemistry program. I applied to Yale. And on a lark, I applied to University of California at Berkeley. And I only applied there, like I was very East Coast. I was like an East Coast person. I was like, I'm not going out to California. That's not my thing. Like I had no interest in going to California. But every 10 years, something called the National Research Council publishes a list of the top graduate universities in the United States. And they had just published their list, and the University of California at Berkeley, on average across all graduate programs, was ranked the #1 graduate school in the United States. And so when I saw that, I was like, well, I might as well apply there, right? If it's #1, maybe I'll get in, and it's, I'll check it out. So I applied, and I think I kind of made it in on the wait list. I just barely sneaked in, but I got accepted, and I went to visit, and let me tell you, it was like, I think within 10 minutes of getting off the airplane, I was like, this is where I'm going. You land in San Francisco and it's like warm out and there's palm trees and you drive over the Bay Bridge and it's just physically beautiful. And I decided to go to Berkeley, which was just an amazing experience. Like probably the 5 best years of my life being a graduate student there. I absolutely loved being a graduate student. You don't have to take many classes. You're working at the bench doing experiments full time. And I love doing experiments. And I'm a good experimentalist. And I got good as an experimentalist. And I love the problem solving of it. I love the working with your hands aspect of it. I love the intellectual challenge of it. But the thing about Berkeley that was amazing were the people I met there. In particular, my advisor, who was just an amazing person. There were no limits with him, right? There was nothing you couldn't do. And I remember I would complain like, oh, this is not possible. And he would always come back and he would say, no, everything is possible, it just hasn't been done yet. And it's just a matter of time before somebody figures out how to do it. And if you reflect, it's actually not so crazy. I mean, imagine somebody who was born in 1900, who lived 80 years until 1980. When they were born, nobody had ever flown yet. The Wright brothers had not appeared on the scene yet. Yet that person, by the time they died, would probably step into a titanium tube with 250 other people and fly at 30,000 feet, 650 miles an hour across the Atlantic Ocean or across the United States. And if you told that person in 1905, and they're 5 years old, that this was going to happen, they would look at you like you were a crazy person, and with good reason. And so, the thing I learned at Berkeley, working with just extraordinary people, was that, if you put your mind to something, the things that can be achieved are just, in many ways, can be limitless. And so here's my advisor. We were on a trip at a meeting in China, and Randy is the one in the middle, and that's me on the left there. I had a beard at the time. And we just saw this placard, which we thought was so perfect. Teacher and student search for theory. And so we decided to take a picture in front of that. The guy on the right, it was another ex-member of, he was another ex-member of Randy's lab that I've known for many years. This is what I did as a graduate student. My Ph.D. thesis title is there. What all that means is that cells make proteins in the cytoplasm of the cell, and those proteins end up in different locations. Some of them are placed into the membrane of the cell. Other ones are actually secreted to the outside of the cell. Like all the proteins in your blood are secreted from cells into your blood, like antibodies, for example. Now, proteins can't spontaneously cross a membrane. They can't spontaneously enter or cross a membrane. They need to have portals in the membrane to enable that. Well, when I started graduate school, those portals, that portal wasn't known. It was known that there had to be one. Actually, that was even controversial because some physicists argued that it could happen spontaneously. But other people argued, no, you need a portal. And I believed in the argument that you needed a portal. And so I developed a genetic screen in yeast, the same yeast you brew beer with and make bread with, Saccharomyces cerevisiae, baker's yeast. I developed a genetic screen to discover that portal. And in fact, I discovered it, I named it, I called it Sec61. This is an image from 2015. So I discovered Sec61 in 1985. So this is 30 years later. This is a three-dimensional structure of the Sec61. It's that cyan protein. It's sitting in the membrane, which is gray. And here's a ribosome that's making a protein. And as it's making the protein, it's threading it through that channel into the endoplasmic reticulum. And that protein will eventually leave the cell and go outside the cell. Now, I did all this in yeast, but these are the human proteins. Because yeast is a eukaryote, just like we are, and the processes, the proteins that exist in a yeast cell to carry out the fundamental biology of cells, all of them have counterparts in human that are very, very similar because they're preserved through the process of evolution. And so that's what I did as a graduate student. Randy went on to bigger and better things. This is a picture I took in 2013 in Stockholm. And this is the King of Sweden. That's the Queen of Sweden. These are various important Swedish people. And this is Randy receiving the Nobel Prize in Physiology and Medicine. Now, after I finished working with Randy, I went on to what's called a postdoc. I went to University of California, San Francisco, which is a medical school of the University of California system. And I worked with this guy, Mark Kirshner, who most recently was chairman of a department at Harvard Medical School. I then took the position of assistant professor at California Institute of Technology. Caltech isn't all that well known on the East Coast. It's a very small school. It has only about 270 faculty, only about 800 undergraduate students. So a very small school, undergraduate school. Yet while I was there for several years, there's an organization in UK called the Times Higher Education of London, and they rank world universities. And for several years while I was at Caltech, Caltech displaced Harvard as the #1 ranked university in the world. So it's very small, very focused on science, very focused on excellence. It was an amazing, amazing place to work. It's like a citadel of science. And so I was there from 1994. In 2000, I became part of the Howard Hughes Medical Institute, which is a funding organization. I remained at Caltech as a faculty member. And now I'm going to transition a little bit, tell you about the science that I did. But I'm going to do this. I know that many of you are not scientists, and I'm going to do this at a pretty basic level. So we're humans. This is my wife, my son, taken on Cadillac Mountain, Acadia National Park. I don't know, maybe my son looks like he's, 7 years old or so. It's probably 18, 19 years ago. We're made of cells. Cells are very small. That's a human hair. It takes, on average, maybe 4 cells to span a human hair. So they're tiny. You can't see them by eye. And there's a ton of them, right? That's, I think, 100 trillion. That number there, 100 trillion, 100 trillion cells in your body. So there's a lot. More than the US debt. All right, so cells are made of molecules, and the molecules, of course, are a lot smaller than cells. So this particular molecule here is a protein. That's a three-dimensional structure bound to DNA, and that spans 11 nanometers. Now, a nanometer is a billionth of a meter. A meter is about a yard, similar to a yard, 39 inches instead of 36 inches. So a billionth of that, so 11 billionths of that is a molecule. To span a hair, you'd need to have 5,000 of those arranged end to end. So proteins are very small. All right, now proteins do most of the jobs in a cell. So one job is movement. Contracting of muscle is movement. That's two proteins that slide against each other, actin and myosin. That's how muscle contracts. Proteins also provide the beams and girders. They're like rebar and concrete of cells. Here you have a neuron, which is a very specialized cell type in your brain with very specialized shape. And its shape is being given by two proteins, actin in red and tubulin in green. Finally, proteins, they're like the workers on an assembly line. Here we have workers building a car. Well, you can have proteins working on an assembly line building other molecules, right? So these are all the various types of things proteins do. Now, when I was at UCSF at the medical school in San Francisco, and when I started my lab at Caltech, I was passionately interested in how yeast cells divide, how they make the decision that they're going to divide to go from one cell to two cell. And the first thing they have to do is they make that decision, number one, and then after they've made that decision, they begin copying their DNA, doubling their DNA, so that they can send one copy of the genome to each of the two daughter cells that are produced. And this had been studied through genetics. The guy who made this figure, Lee Hartwell, first author of that paper, won a Nobel Prize for this work in the early 90s. And he had identified a bunch of genes through genetics that are required for this process of dividing. And I was particularly interested in this early part called start, where cells make the decision, yes, I'm going to divide, and now I need to start copying my DNA. And I wanted to know mechanistically, so this tells you these genes are required, but they don't tell you why they're required. So it's like saying, yeah, you know, for my car to work, I need a steering wheel, and I need an engine, and I need gas, and I need, or electric, depending on your car, and I need wheels, and, you know, intuitively we know what they do, but this is all happening in things that we can't see. So we know they're required, but we don't know what they do, right? And I was seeking to find out how these things worked. And so over the next few years, my lab worked out this scheme. I'm not going to take you in any detail through this in the interest of time, but we figured out that these proteins, essentially what they do is they allow the switching on of another protein that turns on the copying of DNA. Now I'm going to switch from life inflection points to career inflection points. My first career inflection point was while we were doing this work, I'd started the work trying to understand how yeast cells divide, how they make that decision that they're going to divide. And in doing the work, we discovered an enzyme that is involved in degrading proteins. So a key decision point in dividing is there's a protein that blocks the process of division. And the cell destroys that protein. It literally destroys it. It degrades it. So now that blocking protein is gone, and now the cell can divide. And I was fascinated by that as a control mechanism, that there would be things that their whole job is to stop something from happening, and the cell's way to make it happen is to destroy that thing. And I was like, well, that's pretty interesting. And I discovered an enzyme that enables that. And so I completely switched my whole research program to study that enzyme and related enzymes to understand that process of destruction. And how is it controlled? Which proteins get destroyed? How do they know which proteins to destroy? And so forth. Now, the other key inflection point that happened right after that is up until this time, I was really focused purely on fundamental biology. Like, I was just interested to understand how the world works at the microscopic level. And I really didn't care about applications. I had no interest in application. I was just passionate about the fundamental mysteries of life and how it worked. And then I met a person I'll show in the next slide who was much more translational in orientation. And my interaction with him started changing how I thought about stuff. So this is Craig Cruz. He's a professor at Yale. He's been at Yale ever since, the early 90s. I think he started in 96. And we met in 1998 at this location. We both had a fellowship, and the fellowship had a meeting every year, and it was in Blaine, Washington on the Puget Sound. I mean, this is just an incredible place. And we met there and started talking, and we came up with this idea of coming up with new ways to make drugs. And to give you a little bit of background, so most drugs, here, this is an example of a protein. It's obviously a cartoon. This would be an enzyme that degrades things, it chews things up, chews up other proteins. Now, the way most drugs work, they're a lot smaller than a protein. A drug might be 100 times smaller than a protein. But as in the example here, if you stick a little bar in the mouth of this vicious Pac-Man, you're going to basically block the action of the Pac-Man. It's not going to be able to close its mouth anymore. Well, a drug is very similar. It's much smaller than the protein, but it fits in the business end of the protein, in this case, the mouth, and blocks it from doing its thing. So that's how most drugs work. So cells, I've already described to you, have this mechanism of destroying proteins. And it can destroy proteins for different reasons. One was, I told you about the protein that blocks cell division, and you have to get rid of it to divide. But there's also proteins that are constantly going bad in cells. And somebody asked me yesterday, do I use AI in my work? That image was generated by an AI prompt in ChatGPT, and it was perfect, exactly what I wanted. So when you're in your house, you open the fridge, and if you haven't been around for like 2 weeks and you forgot to empty the fridge before you left, you come back and you'll notice that some things have gone on that are not exactly pleasant. Like some food has gone bad. And you can tell it's gone bad. You can see that it's gone bad. You can smell that it's gone bad. So you grab that food and you throw it in the garbage. Well, cells have similar mechanisms of recognizing proteins that have gone bad. They become oxidized, perhaps, and recognizes them and destroys them. And here's an example. I've already told you about, I think I mentioned ubiquitin ligase. These are enzymes that initiate this whole process. And normally, they have to recognize their proteins through some surface complementarity. Now, the example I've shown here, these proteins don't have surface complementarity. One has a rectangle thing and the other one has a triangle opening, so they're not going to fit together. But that normal protein may have undergone some damage, which now creates a rectangle opening that this can fit into. And so now the ubiquitin ligase can bind that protein, and what it does is it transfers this protein ubiquitin to it. Now, ubiquitin is a signal for a large Pac-Man, vicious Pac-Man like I had in the other slide, an enzyme that chews other proteins up. If you have ubiquitin on you, you get chewed up and the protein gets destroyed. And so it's equivalent. The human there is the ubiquitin ligase, the damaged proteins are the food in the fridge, proteasome is like the garbage pail. And so the idea we came up with was, can we repurpose a situation like this where there's no complementarity? Can we make an adapter molecule that would enable these two to nevertheless come together so that we could specify the elimination of any protein at will by making an adapter molecule? And it turns out that works, and it's launched an entire industry in drug development. And so with that, I'm just going to skip, I'm near the end anyway, I'm going to skip right to the end and just, reflect on, like for me, the important things in my life were having mentors and surrounding myself with the best people, because there's just nothing like the people around you are so important to you, and surrounding yourself with the best possible people is so important. It was for me. Number two, diverse experience. Like, if you have a chance to have a new, different experience, take it. I've learned so much from diversity of experience, of taking risks, of doing different things. I've always learned from doing those things, and it's really enriched me, and it's led to unexpected opportunities. Number three, my parents emphasized my father never went beyond 5th grade, emphasized education, education, education. I don't care what form your education takes, but lifelong learning, I think, absolutely critical. Number four, and I think my collaboration with Craig Cruz underscores this, you can get more done working with other people than you can get done yourself. And so, for me, seeking out people who complement what I know has enabled me to do things I could have never done on my own. Positive attitude. A lot of people in the world are pessimists. It's not hard to find pessimists. I found that, most people who are successful are optimists. And I've always been a very optimistic person. And having a positive attitude, if you believe something can be done, there's a much better chance that you'll get it done. And finally, exercising all these things. Look, I think you guys are in a great position. This is a great school. It prepared me very well for my life. Those of you in science have a fantastic mentor in Char. And, I think the potential for you, the world is a huge place. Many of you will make your lives in Waterbury, but some of you will make your lives elsewhere. And I can tell you the world is a big place. There's a lot of different opportunities out there that being in Waterbury you don't even know exist. Like, I didn't know the whole world of biomedical research existed. And so, my advice to you is keep an open mind, seize opportunity, and keep learning. Thank you.
Thank you, Dr. Deshaies. Community period is just about to end. If you're in the Science Academy, you can stick around, and the rest of you will be dismissed when the bell rings. But thank you very much. This is incredible. So thanks again.